Okay, this is the podcast for STEM biology, chapter 23. Yes, we are going backward a chapter. We did chapter 24 with viruses. Now we're doing chapter 23 with bacteria. There are 3 sections, and we're going to cover section 1 and part of section 2, break, and then do part B in a separate podcast. What's pictured here is bacteria, by the way. Okay, first thing we're going to talk about are domains. There are 3 domains of living organisms, which means if you take every living organism that's on the earth, you can classify them into 3 domains. Organisms are either prokaryotes or eukaryotes, and that kind of coincides with what domain they are. For us, we are eukaryotes, and so we are in the domain eukarya. There is only one domain for that type of organism. In prokaryotes, however, which is what we're studying now, there are 2 domains, domain bacteria and domain archaea. That's how you say it, archaea. And notice that all of organisms they came from a common ancestor, the first cells. Okay, so there are 2 major domains of bacteria. First of all, make sure you understand what a domain is. A domain is basically a grouping, a classification. So there are 2 main groupings or domains of bacteria, and that is archaea and bacteria. So let's go through each one of those and describe different organisms and what they do. Let's do archaea first. Archaea are different than the regular bacteria. Archaea are not bacteria that you would find every day around. They differ, and the main thing they differ in is their composition and what's inside them. Their cell wall composition is different. There are different amino acids in there, different types of lipids in the cell membrane. Okay. Also location. Archaea are found in extreme environments, places that you would never, really see in an everyday life, such as swamps, salt lakes, and hot springs. There are 3 main groups of archaea that we're going to learn about. First group are the methanogens. These are bacteria that convert hydrogen gas and carbon dioxide into methane gas. For these type of bacteria, oxygen is poisonous, and so they are only anaerobic. And they live in anaerobic environments like deep fresh water, the marine mud down at the bottom of the ocean, swamp mud, and sewage. Methane is produced by them and bubbled out, and you might have heard the term marsh gas. Archaea, group number 1, the methanogens, are also found in the intestines of cows and termites. You might have seen cows belch or whatever in the pictures of them. What they're belching is methane because they're producing methane in their intestines. And it's under research of how all this methane that they're belching out is affecting the environment. Okay, so this is what methane's formula is. It's a carbon with 4 hydrogens attached. This is what the methanogens look like under a microscope. And that's a picture of a cow belching methane. All right, group number 2 are the halophiles. Philes means love, halo means salt. So these are also prokaryotic cells. Well, all of them are for this chapter. And these are bacteria that are salt lovers. They live in environments that have a very high salt concentration, such as the Great Salt Lake and the Dead Sea. And you might have learned about those in history or some kind of social studies class. Normally, if you have bacteria and you put them in a lot of salt, it's going to kill them. But not these guys. These guys, normally, they thrive, they want to. They have adapted to this high salt concentrations, and it won't kill them. So this is what they look like under the microscope. And the Great Salt Lake is in Utah, right there. That's where you would find them. And they're found in the Dead Sea, which is by Jordan and Israel. All right, archaea group number 3 are the thermoacidophiles. Thermo has to do with temperature, and then acido is talking about acidic, and that's what they like. So they live in acidic environments that have a very high temperature, such as the hot springs at Yellowstone National Park. They can live up to 110 degrees Celsius and at a pH of less than 2, which is very acidic. Normal bacteria cannot live in that hot of a temperature. You boil water, tell you to boil your water before you drink it in certain areas, or when the water supplies go bad, and normal bacteria won't survive it. You're okay, you can drink it after that, but not these guys. They also live near volcanic vents on land or near hydrothermal vents called black smokers, which are cracks in the ocean floor. Okay, so these guys look like this. There's Yellowstone National Park. You would find them in the hot springs there. And then this is showing you the cracks in the ocean floor and what a black smoker is, and that's where you can find them as well. Okay, so that finishes the domain Archaea. Let's do the domain Bacteria. They're prokaryotic. These are the common bacteria you would find every day because you would see them, or you wouldn't see them, but you find them in the soil, in our bodies, and they live symbiotically in other organisms. They occur in many shapes and sizes, just like viruses. They have three basic shapes. A rod-shaped bacteria is called bacilli, and that's what E. coli is. They also can be circular shaped, and that's called cocci. If they are circular shaped and they're in a chain, so it's a circle linked to a circle linked to a circle, that's called streptococci. When they're in clumps, they're called staphylococci. And then they also can be spiral shaped, which is called spirilla. So here is bacillus, rod-shaped bacteria. Here is coccus. Notice that these ones are staphylococcus because they're in clumps. That's very similar to the ones you saw in the prokaryotic slide in the microscope lab, the cell lab. And then you have your spirillums, which are spiral shaped. Okay. There is a procedure called a gram stain that you can do on bacteria. Doctors do this all the time. You're sick, you have a sore throat, you go to the doctor, they swab your throat, you gag. They take the swab, they say, we'll be back in 5-10 minutes. They come back and they know exactly what you have and they know exactly what kind of antibiotic to give you. That's because they are doing a gram stain. Basically, they're looking at the structure of the cell wall of the bacteria that you have the infection. And so they look at the cell wall, they know what kind of cell wall it is, and then they can give you the right antibiotic. So bacteria fall into two categories, gram stain positive or gram stain negative. If they are gram positive, these bacteria have a thick layer of peptidoglycan in their cell wall. Peptidoglycan, I'll show you what it looks like in a minute. When you see gram-positive bacteria under the microscope, they are purple. Gram-negative bacteria have a thin layer of peptidoglycan in their cell wall, and they appear reddish-pink under the microscope. Okay, so what is peptidoglycan? Peptidoglycan is a protein-carbohydrate mix. Okay, you see the protein part, and then you have your sugar part right here. So it's a protein-carbohydrate mix. Pretty big molecule, and it's found in bacteria cell walls, but not Archaea. Archaeas do not have peptidoglycan in their cell walls. So some bacteria have a thick amount of this compound, and some bacteria have a thin amount. If they have a thick peptidoglycan cell wall, then they are gram-positive bacteria, and they appear purple under the microscope. If they have a thin layer of peptidoglycan in their cell wall, notice that they actually have a membrane on the outside of their cell wall because this is so thin. They are gram-negative and they appear pinkish red under the microscope. Okay, that is called the gram stain. This is how you do a gram stain.
Eubacteria are categorized into two groups based on their response to a laboratory technique called the gram stain. The two groups are called gram-positive and gram-negative. In the first step of the gram stain, a colony of bacteria is smeared on a slide and fixed onto the slide with heat. The slide with the bacteria fixed onto it is flooded with a purple dye called crystal violet. All of the bacterial cells are stained purple after this step. Next, the slide is flooded with iodine. The iodine binds the crystal violet to the cell walls of gram-positive bacteria. All of the bacterial cells are still stained purple after this step. Next, the slide is rinsed with alcohol. This removes the color from gram-negative cells. At this point, the gram-positive cells are purple and the gram-negative cells are colorless. Finally, the slide is rinsed with a pink dye called safranin. The safranin stains the gram-negative cells pink so that they can be seen under a microscope. When the gram stain process is complete, the gram-positive cells are purple and the gram-negative cells are pink. Gram-positive bacteria retain the purple gram stain because they have more peptidoglycan in their cell wall than do gram-negative bacteria.
Okay. All right, now we're going to start important bacterial groups. Bacteria are classified based on their properties, their biochemical properties, and their relationships. There are 5 main groups we're going to go through. They are important, not so important as to the names of them, like, because I'm going to give you a lot of scientific names, but to know that in general, like whether they're, good bacteria or bad bacteria that cause disease, and which diseases are bacterial. Okay, so the first one we're going to do before we go to podcast part 2 is the Proteobacteria. We do this one first because it is the largest group and it's most diverse group. Most of Proteobacteria live symbiotically with other organisms. So what does that mean? They live with another organism and they both benefit. Okay? This bacteria, called Rhizobium, is a bacteria that live in the nodules inside the roots of peas and beans, like they're called legumes. And what they do is they fix nitrogen. Okay? What does that mean? Our atmosphere is 79% nitrogen. And so the problem with that, however, is that we can't use that nitrogen at all. Animals can't use it, plants can't use it. So this bacteria take the nitrogen in the air and change it to ammonia. And that process is called nitrogen fixation. And it's part of the nitrogen cycle. And so that's important because ammonia plants can use, but plants can't use the nitrogen gas that's in the air. So if plants can use ammonia, then they can make their proteins and their DNA, and then we eat plants, and that's how we get our nitrogen. So it's important. That's why it's important to eat vegetables. So that is an example of Proteobacteria, that they live symbiotically with other organisms, and they fix nitrogen to a usable form. Here's a picture of the roots of legumes and peas, and so they would be found on these roots. And this is a picture of the nitrogen cycle. The nitrogen in the atmosphere is taken by nitrogen-fixing bacteria like Rhizobium and then made into ammonia, which is then used for plants and animals. All right, but not all Proteobacteria are good bacteria. Some cause diseases in plants and animals. Some causes tumors in plants, like Agrobacterium. Some cause Rocky-mounted spotted fever in humans. Some cause stomach ulcers. So these are examples. You don't have to know these specific names. They might show up on a worksheet or a review, but they're not going to be tested on them. Another one, some Proteobacteria are enteric, which means they live in our intestines. So this is our intestines here, and there you can see that there's bacteria living in there. E. coli can be a good bacteria. Some strains of E. coli make vitamins inside our body, and they help digest our food, or help with digestive enzymes in there. Some E. coli, however, cause food poisoning with salmonella. So these are all Proteobacteria. They cause foodborne illnesses that invade our cells and make toxins, which makes us sick and want to throw up. Okay, and we're going to stop here for podcast 1.